Welcome to our journal club. The paper that we're presenting today is called VTA to basal amygdala dopamine projection contributes to signal selling somatosensory events during fear learning. And it is about the role of dopamine neurons in fear learning. However, we will be focusing on figure 2, specifically from A to F of this paper. So the main or the big question for this paper is how does fear learning occur in animals? However, why is it important? The researchers want to address this because discovered information might give more insights into anxiety problems and post-traumatic stress disorders in humans. And one of the main issues of this psychological disorders is maladaptive plasticity, and dopamine projection possibly contributes to a teaching signal for plasticity. By understanding how dopamine projection works and how it is different from people with such problems as anxiety and PTSD, new solutions for treating these disorders might arise.
This is the conclusion of the previous figure. With the help of dye, specifically CTB Alexa 647, which was injected into the BA of DAT mice, BA projectors could be found in the VTA. The results showed that more BA projectors were found in the dorsal half of the VTA compared to the ventral half. But no BA projectors were found in the lateral and in the posterior parts of the VTA.
Okay, so we are going to be specifically focusing on figures 2A through F. The main research question designed for relevant experiments of the figure, and the main conclusion that they came to was VTA neurons respond to foot shocks and therefore acquire a conditioned stimuli response. Their main question to conducting all of these experiments is, what exactly is the role of the VTA neuron in fear-based learning? And with their question, their objective for conducting all of these experiments in figures 2A through F was to investigate whether VTA neurons induced a conditioned response during fear-based learning. In regards to their approach, while they used multiple, the main approach was for tracing techniques of DAT plus dopamine neurons in the VTA. They started off using optogenetic methods with retrograde virus, which in simpler terms, optogenetic methods are biological techniques used to observe and control the activity of neurons with the light, and that light then manipulates neural activity. They also used in vivo optrode recordings, and that they use optrode implantations, which consist of 4 tetrode bundles, which then lay atop or around of the central optical fibers. And they do that in order to find extracellular spikes in the neurons. They also used foot shocks and tones, and they did so in order to observe the physical reaction that the mus musculus had when conducting the experiment with fear-based learning.
This is figure 2A, and it shows the responses of mice to tone blocks and foot shocks. The mice went through 3 phases, which were each 1 day long. The first day is called habituation phase, the second day is called training phase, and the third day is called cued retrieval phase. Each day had 6 tone blocks. Day 2 also had 6 1-second long foot shocks, which were paired to the 6 tone blocks of that day. The mice responded to the tone blocks and foot shocks in freezing. The a-axis of the graph shows the number of tone blocks, and the y-axis shows the freezing level of the mice. the first tone block on day 1 triggered a relatively high freezing level, but throughout the day, the freezing level decreased rapidly, which means that the other 5 tone blocks weren't able to increase the freezing to the same level as the first tone block. On day 2, the tone blocks were paired with foot shocks. Therefore, the freezing of the mice increased rapidly to a very high level. On day 3, the foot shocks were removed again, but the freezing level was even slightly higher than on the second day. The high level on day 3 is caused by fear learning.
The figure 2b and c are presenting to us one of the techniques used in the experiment, which is the implantation of an optrode with 16 recording channels into the VTA. It was done to see the neural circuits and detect extracellular spikes. Figure 2b is showing the schematic optrode implantation in VTA of the amygdala. The optrode implanted here was 473 nanometers, and figure 2c shows a post hoc histologic image where optrode is dashed line, as we can see here, and tetrodes are shown with arrows. And at the bottom, there's a scheme of arrangement of 4 tetrodes around the optical fiber. These techniques were used to try to optogenetically identify the BA projectors in the amygdala. However, results were not much helpful, and it was assumed that it was due to the big amount of BA projectors in the VTA. Which is why mouse was put under DAT promoter so that the researchers would be able to identify the fraction of recorded units. And figure 2D is an illustration of optogenetic identification of DAT plus units in VTA before, here, and after the light pulse activation. The observed data here shows that from 2 to 8 milliseconds after the light pulse, the spike clustering was collected, and that was later used as a control group so that it can always be subtracted from the other spike clusterings during training day and retrieval day. This specifically was done because the first clustering was due to the light and not to the conditioned and unconditioned stimuli.
All right, moving on to figures 2E through F, the researchers mainly wanted to measure the DAT dopamine neuron spikes on both a training day, seen in figure E, and a retrieval day, seen in figure F. Furthermore, explaining the axis, we're going to have frequency on top and we're going to have z-score on the bottom. Frequency is going to be the measurement of the action potential firing of the dopamine neurons seen in the mus musculus. And the z-score is going to be the measurement of the mean or the average in standard deviations. So on the bottom of the frequency, we're going to see the average over our n equals 30 tone presentations for each tone block. And on the right-hand side of figure E, we're going to see the average of the n equals 6 foot shock presentations. Now, focusing specifically on figure E, which was our training day, this was the day that foot shock stimulation was paired with our tone blocks, our conditioned stimuli. Researchers were able to notice that there was a developed short latency response, which in other words states that there was an early peak above the threshold. And moving forward to figure F, researchers were able to notably see that the response was maintained on the retrieval day. So there was an increased spike with the tone blocks despite the removal of the foot shock stimulation. So with this, researchers were able to conclude that there was an increase in action potential firing, and this shows that there was an acquired AP firing response to our conditioned stimuli in fear memory. So to recap our conclusions, freezing level in mus musculus increased to conditioned stimuli, our tone blocks, despite the fact that we removed the unconditioned stimuli, the foot shocks, showing that VTA neurons, including dopamine, do play a significant role in auditory cued fear memory formation. Another conclusion that goes hand in hand with this is that the response to foot shock pain observed in mus musculus was the freezing. And another conclusion they were able to make was that a great majority of VTA units were present in fear-based learning. And within those units, they found the DAT dopamine neurons. And they both further responded to an increased firing due to the foot shocks.
And we had a couple of questions towards the paper. The first one is in the optogenetic part of the experiment, the researchers were not able initially identify the BA projectors in the VTA, and they just suggested that it was due to the big amount of projectors in the amygdala, which is why the mouse later was put under DAT promoter. However, despite the results not being fruitful, the researchers still used the herpes simplex virus as a retrograde virus to identify BA projectors, or did they remove it completely and moved forward? And if it was still used, then what was the reason? And the second question was why were the foot shocks exactly 1 second and not less or more? Was there a specific reason for this timing? And this is our references. Thank you for listening. This was our presentation.